Today we'll have an excellent research talk. I want to remind or let people know of some of the recent research successes that we have had in the department. You may or may not have, this is through the efforts of faculty. These are not related to stimulus dollars, but Dr. Michael Helmuth received an R1 grant, sort of the major independent grant type of grants. Dr. Bruce Cairns received an R1 grant, and recently the T32 grant, which is a trauma training grant, was also, we received verbal confirmation that it was approved. The commitment and interest to research it remains strong in the department, even in some of the trying times. And today we'll hear some more about research in the Department of Surgery. Dr. Lesley Marson, who is a research professor in the Division of Urology, will let her introduce the rest of the presentation today. Dr. Marson.
Good morning. Okay, I'm going to talk today about neural paths regulating pelvic crosstalk. I'm going to present first, and then Andrew Dobberfuhl, a student that's been working in my lab for a year, will present the work that he's been doing, and he's done a tremendous amount of work. This is his fourth presentation, I believe, this year already, so I've been going to sorely miss him, but he's done a great job. So without further ado, we'll move on. I just wanted to share this with you, and maybe this will help you wake up, get those neurons firing. This is an email that Suzanne received from a faculty that wouldn't be here today, but I'm sorry to say that we're going to talk about a single pelvis today. So why understand the neural control of pelvic organs? Pelvic cross-sensitization is thought to be one of the major factors that results in chronic pelvic pain. And as you're all many surgeons here, you know that surgical intervention can often damage nerves that can result in various dysfunction. Also, trauma, infection, diseases really do affect the pelvic organs, even if the disease is in the brain. So the outline of the talk today, I'm going to give you an overview on pelvic crosstalk, which is an area of interest of mine, and then Andrew is going to give you some detailed studies that he's conducted in the lab, as I said earlier. So the pelvic organs share sensory neural paths, and these include the hypogastric, pelvic, splanchnic, and pudendal nerves. They also share the main efferent neural paths, and also they share brain and spinal pathways. And that's a major interest of mine. So I'm just going to show you this so that you can see this is sort of the pelvic organs of the female. And you can see the bladder, the clitoris, and the vagina, and how closely associated these organs are with each other, and the rectum, pelvic floor. And these pelvic organs subserve multiple functions. And this is a male. And again, a lot of different organs doing a lot of different things very closely related together. And a lot of the chronic pelvic pain is manifested more in females than males, but obviously both sexes fall into this syndrome. So I'm going to just overview with you the three different branches, or the three different ways pelvic pain can be developed through the periphery, spinal, and the brain pathways. I'm going to use a review here, and the references here is quite a recent review which summarizes pelvic organ cross-sensitization, and if you're interested, this would be a good one to read. So one possible way of causing hypersensitivity is through the dorsal root ganglia. and the diseased organ gets inflamed or injured, and you get activation of neurons. If you just look at the red path, the hypersensitization and hyperpolarization within the dorsal root neurons. And some of these neurons, a single neuron, innervates multiple organs. And then you can get an axon reflex that will go to an adjacent organ, which will produce instability within that organ. There's some evidence for this. There's not a huge amount of evidence, but this is one definite possibility that needs to be bore in mind when we're thinking of what causes this crosstalk between organs. There's a convergence of afferent information in the spinal cord, and there's a lot more evidence for this. Here we see the root here in blue. Now we have, again, the diseased or traumatized organ here, and you get inflammation here. Of course, it goes through the dorsal root ganglia into the dorsal horn, and then it synapses on interneurons within the intermediate gray, medial gray, and lateral cell column, which changes the activity of these cells and these gating mechanisms of the cells, as well as the tonic firing rate. And then you get a change in outflow. And then again, that can cause a disease or inflammation or instability of an organ. And most of the work in this field has been done pretty much on colon and bladder. And a lot of the work has been done in rats and cats to try and look at the areas in the spinal cord or the neurons in the spinal cord that are regulating and mediating this dysfunction. And the first point I have here is, this is from neurophysiology studies where they record from either a single neuron or a group of neurons within the spinal cord. And if they distend the colon and or the bladder, these neurons fire together. So this shows you here that about 26% of the recorded neurons in the dorsal horn respond to both colon and the bladder. Deeper in the spinal cord, there's a larger proportion, and these are mostly interneurons, so there's a lot of connectivity within the spinal cord. Also, studies have been looked at that have looked at interaction between the uterus and the colon, and again, there's a large percentage of overlap within these various regions of the spinal cord, mainly the thoracic, lumbar, cervical regions. So also, studies that we've done mostly have been neuroanatomical studies. These neurophysiological studies are really difficult to do and take a huge amount of time. There's pretty much 2 or 3 groups that work on this. But we have shown that common interneurons are activated by the various pelvic nerves in the spinal cord, and also that they have common efferent projections to multiple organs. So the next step is, or the next possibility is convergence of information in the brain. And this is shown here in the green pathway, if you follow that. And again, sensory activity from the diseased organ, and this could be the bladder, it could be the instability of the skeletal muscles, it could be the vagina, it could be the penis, causes changes in the neural activity mechanisms and interconnections within the spinal cord, which send projections up to the brain. And there are a variety of possible areas for convergence of sensory information in the brain. This includes the gracile nucleus, the nucleus of the solitary tract, the reticular formation, the thalamus, the cortex, and as well as many others. And then there are efferent projections then that communicate back down to the spinal cord that changes the efferent outflow and the responses to these organs. Another good paper for you to look at is that by Dr. de Groot, and he's basically the guru of the bladder world. And This is just taken from a paper that he wrote in 2006. This is his sort of famous picture that he shows in his talk. So, but what you can see here is, and he works on the lower urinary tract, is that there's a lot of integration of the sympathetic pathways. Here's the sympathetic chain, and which then goes down through the mesenteric ganglia, through the hypogastric nerve to the pelvic plexus. You've also got the pelvic nerve coming down that also receives fibers coming from the sympathetic chain as well from the spinal cord that go into the pelvic plexus. And then you've got the postganglionic or post neurons, that cavernous nerve, for example, that goes to the penis, coming from these ganglia. So you've got the possibility of a lot of interaction here out in the peripheral area as well. So, you know, when you do your surgical interventions, it's important to know where these are. And also then you've got the sphincter muscles that go through the pudendal nerve and back into the spinal cord. I like to show this picture again from this paper because this shows you some of the basic science that's done and how we can build up a picture of where these inputs interact. And Andy will show you some of this a little bit later. But this just shows you an example of cells, neurons that were activated with CFOS. This is a media early gene, and what they do in their studies is they instill acetic acid or other toxic chemicals into the bladder and look at the cells that are activated in the spinal cord. We do other things, but more physiological things. We evoke erectile ejaculatory responses and then activated cells in the spinal cord in the brain to see which ones are regulating and mediating those sorts of reflexes. But also you can see the bladder afferents that are coming in here labeled with a HRP tracer, and then also the pseudorabies virus tracing, which is a useful way to look at transsynaptic connections within the central nervous system. And what you can see here is that there's a lot of overlapping areas, especially in the medial cord and IML. So there's a possibility of these are the same or interconnecting pathways. So what we've done in our lab, and this was done with the help of Jessica Weedi, who was originally a biology student here at UNC, and she's now gone into dentistry, but she helped us do one of the studies where we stimulated the pudendal nerve and the pelvic nerve of rats, and then we looked at which areas of the spinal cord were activated by these two different nerve stimulations, and this is a somatic nerve and then the parasympathetic nerve. And I'm just going to give you the overview of this, but what you can see is there's overlapping activity, and just this is a stimulus gradient where we counted the CFOS cells, and the darker color shows the more cells in the dorsal horn, as well as a large area in the medial gray that shows you overlapping inputs of these two different branches of the nervous system. Some of the other studies that we do, we integrate the CFOS mapping of activated neurons with mapping of efferent neurons using the pseudorabies virus. And again, you can see here, this is the pudendal nerve stimulation, so this is the afferent branch, and this is the efferent branch where we put a virus into the skeletal muscles, goes in, of course, through the pudendal motor neurons, and then it makes connections in the spinal cord, and Andy's going to show you some of the work on that later. But again, you can see some of these things are overlapping. So I'm going to hand this over to Andy. He's going to give you the nuts and bolts of some of the stuff that he's done.
Good morning. My name is Andrew Dobberfuhl. I'm a fourth-year medical student, as I've already been introduced, and a Howard Holderness Fellow between my third and fourth year of school. So today I'm going to talk to you about elucidating neural sarcos. So to start, it'd be appropriate to tell you a little bit about the history. I'm sure everyone's ready for a history lesson right now. So around the turn of the century, there were two opposing theories of how the brain worked. One was upheld by Golgi, the reticularist theory that the brain was one single network, and this was opposed by Cajal's theory that supported the neuron doctrine, which we now use today, that neurons are single cells that synapse with each other. So to study neural circuits, there were two methods used. One was retrograde degeneration, where targets were ablated and then subsequently studied, as well as anterograde degeneration, where axons were lesioned and then studied. Together, well, the biggest problem during this time was labeling a single neuron, because antibodies against a single target weren't introduced yet, and Golgi discovered that you could use silver impregnation, which would randomly, which to this day we don't know why, but it would randomly label one in approximately every 100 neurons. And then Cajal used his technique to first identify neurons, and together they won the 1906 Nobel Prize for their study of the nervous system. So in the 40s, the anterograde degeneration techniques were improved and longer projections were elucidated. And then around the 70s, as Lesley mentioned, the introduction of horseradish peroxidase allowed in vivo transport of this for the study of retrograde and anterograde projections, as well as when conjugated with wheat germ agglutinin, you could study transsynaptic connections. And around this time, radio-labeled amino acids were used for autoradiography. And later, fluorescent dyes were introduced, which allowed multiple connections to be traced simultaneously, as well as some of the newer tracers that we use here in the lab, collar toxin, as well as Phaseus vulgaris, leukogluten, biocytin, and dextran. And then, as Lesley mentioned, the FOS activation with CFOS. This is an immediate early gene, which is rapidly expressed, but it's only transient. And then what we mainly use in our lab are some of the neurotropic viruses. So there are two main families of tracing viruses. One is in the herpes family. This is a DNA virus, a larger genome of 70 genes, and can be transported both retrograde and anterograde, and also be modified easily with DNA manipulation techniques. Other investigators have used the rabies virus, which is a simpler RNA virus with only 5 genes, but only is transported in the retrograde manner. But as we all know, it infects humans and is not as safe. So some advantages of using neurotropic viruses are that we can traverse multisynaptic pathways. And this is really exciting because this allows us to infect one neuron and then see which subsequent neurons are linked to this single neuron. And then also the natural life cycle of the virus allows for amplification of the signal. And then viruses are highly specific to synaptically connected neurons, so they don't infect their neighbors that are not connected by synapses. The only problem is, well, just with the viruses in their unmodified form, is there's a lack of synaptic specificity and inability to target particular cell types, and you really depend on the strength and number of connections, and there's also the natural induction of cell death and lysis, which, so what we use in our lab is a pseudorabies virus. This is a herpes virus, aside from its name. And this was originally pathogenic in the swine industry, discovered about 60 years ago, and it naturally infects, as you can see here, a variety of hosts, but does not infect humans or non-human primates. And around 1961, Bartha discovered the Bartha strain, which is an attenuated version which lacks particular envelope proteins and only is transported in the retrograde manner, and has a much slower spread and reduced cytopathic effect and allows for longer survival times, so we can label more neurons. And then this has subsequently been modified for a green fluorescent protein expression. So just a little background, we're investigating vaginal innervation. and as Dr. Marson mentioned, is innervated by the pelvic, pudendal, and hypogastric nerve, carrying parasympathetic, sympathetics, and somatic nerves. So we injected pseudorabies into the vagina of sexually experienced overactimized female rats, allowed them to survive 4 to 5 days, and then anesthetize them, perfuse the animals, and fix their spinal cords and brains, and then remove these for visualization of the pseudorabies virus. So this is our typical injection site. Over here on the right, you can see the lumen of the vagina, and this is a stratified squamous epithelium. Then here's your smooth muscle bilayer, which was really our target for our injection. In a separate study, we're looking at the levator ani muscle, which along with the ventral bulbar spongiosus muscle. The levator ani muscle is very interesting because it's a hormonally responsive, sexually dimorphic muscle important for erection in the rat. And apart from its name, it's not exactly the same as its human counterpart. So we performed 3 experiments in these animals. First group of 24 animals received pseudorabies into their left levator ani, and then sacrificed 3 to 5 days later. Another group of 7 animals, adults, received fluorogold into their left levator ani, and this is one of our fluorescent dyes. And then they concurrently received collar toxin into their left ventral bulbar spongiosus, and they were sacrificed after 9 days. And in a smaller group of animals, of rat pups only 30 days old, we injected pseudorabies into the bulbar spongiosus and sacrificed after 3 days. So this is our typical injection site. On the left, you can see an adult, and on the right, you can see a 20-day gestation fetal rat. So LA is our levator ani. It forms a partial sling in the rat around the rectum. And then here, adjacent to it is the ventral bulbar spongiosus and the distal bulbar spongiosus. So these were our 2 targets for the collar toxin and fluorogold. And on the right, you can see the 20-day rat. So here's a typical section of the spinal cord. So at L1, the level where you'd find your sympathetic preganglionics, we see labeling in the intermediolateral region of our preganglionic efferents. And then we also see labeling in the medial gray. And then at the level of L6, where you'd find your parasympathetic preganglionics, we see labeling in the medial and intermediate gray of interneurons. So just to demonstrate some variability of infection, because this was from our vagina study, depending on where the neurotropic virus ends up, you will label different neurons. So in A, you see predominantly a sympathetic labeling pattern of T12 to L2. Just to note, these are all individual animals. We have on the x-axis our spinal segment stratified, and on the y-axis a number of neurons counted. And then B shows a much different type of infection with a lot of labeling around L5, suggesting a parasympathetic labeling. And then C is kind of a combination of both. So these are the results of the 18 animals that were injected with pseudorabies. This is comparing 4 and 5 day infection. At the early stage, we see predominantly a parasympathetic labeling pattern with a 5-fold increase at the later infection stage in a more caudal distribution. And then in our levator ani study, we see a similar labeling pattern. Here in the dark blue, again, here we have mean number of cells infected. In dark blue, you can see early on, this is between 3 and 4 days, labeling in the lumbosacral region. And then you have a 5-fold increase in the number of labeled cells. This is just the number of cells stratified over different areas. And we see predominant labeling in the lateral, intermediate, and medial gray. So then results of our fluorogold and collar toxin study, these labeled motor neurons in both the dorsal motor nucleus and the ventral horn in all of our animals, but only in L4 to S2, as these 2 tracers don't cross synapses. And then we found co-localization to both muscles in about half of the animals. So then in our pup, looking at its spinal cord, the labeling was very similar to our adult. And here on the left at L1, we have labeling in the IML. In L4, we see some labeling in the medial gray, and then at L5, we see labeling of the dorsal motor neurons and the dorsal gray commissure. And then moving up to the brainstem in our vagina study, we see labeling of the nucleus tractus solitarius. This is just your caudal medulla. Here's your central canal. And we also see labeling in the raphe obscurus, which is important for cerebellar coordination, the peripyramidal tract, the ventrolateral medulla, as well as at the level of the pons, we see labeling of the Barrington's nucleus of the lateral tegmental area, which is very important for implicated in micturition. So then in the hypothalamus and forebrain, we see labeling in the paraventricular nucleus, which sends direct projections to the lumbosacral parasympathetic motor neurons that we discussed earlier. And this is your optic chiasm. Here in the medial preoptic area, which is important for sexual motivation, we see labeling as well as in the bed nucleus stria terminalis of the forebrain, which is important for sexual identity in humans. We also newly found labeled cells in the suprachiasmatic nucleus, which is important for circadian rhythm. So, and then looking at our levator ani study, we see a very similar labeling pattern. So we see labeling in ventrolateral medulla, which is important for inhibiting the UG reflex in rats, and we also see labeling again in Barrington's, the paraventricular nucleus, and the medial preoptic area. So just a quick summary of everything where we saw labeling in our vagina study. So after pseudorabies is injected into the vagina, it's taken up by the motor neuron end plates and transported through postganglionic neurons through major pelvic ganglia to the thoracolumbar region and the IML and medial gray, as well as the lumbosacral region, makes synapses with the sacral parasympathetic nucleus and dorsal gray commissure. And then multiple synapses are made with interneurons at many levels, and then the brainstem, there are many connections made with the ventrolateral medulla, paraventricular nucleus, raphe obscurus, raphe magnus A5, and the A5 noradreneric group, as well as Barrington's nucleus. Then the midbrain and hypothalamus, we make connections with the nucleus tractus solitarius, the periaqueductal gray, the subquerulus, the locus coerulus, the red nucleus, then posterior and lateral hypothalamus. And then these make synapses with the forebrain, the medial and lateral preoptic area, the bed nucleus, stria terminalis, ventral tegmental area, Edner-Westphal's nucleus, arcuate nucleus, and suprachiasmatic nucleus. So what did we find that differed from other studies? Well, here in the yellow are our main differences from other studies that look at pelvic organs, and we find labeling in the area postrema. This is implicated in the gag reflex. It's been labeled in other studies after injection of pseudorabies into the colon. And we fail to see labeling in the ventral medial hypothalamus, which, as these were overectomized rats, this is an estrogen-dependent area and normally responsible for lordosis behavior in the female. And just to summarize our levator ani study, the rat levator ani is not totally homologous to its human counterpart and is actually a sexually dimorphic dorsal extension of the bulbous spongiosis. And together with the bulbous spongiosis, it forms a muscle complex that's tightly coordinated for erectile function. And as these are two distinctly separate muscles, we've demonstrated that they receive projections from a single motor neuron by co-localization of fluorogolden collotoxin. And then our study of the levator ani is in concordance with other studies of pelvic organs, and by demonstrating our techniques in the pup, these could possibly be used to look at early neural development. So that's pretty much the end of my part of this, and thank you for your attention.
Okay, I'm not going to belabel the different parts of the brain, because unless you're a neuroanatomist, it gets pretty boring, right? Unless you're really good at Latin. So Andy gave you quite a good summary there of what we found, but I think it's sort of important to reiterate, in the spinal cord, what's really becoming apparent in many different labs now is there's a column of cells that transverse multiple segments in the medial grey that really tightly coordinate and integrate the information from different nerves, somatic, parasympathetic and sympathetic nerves, to help coordinate the outflow that we want, whether we want the peeing reflex, micturition, continence, erection, ejaculation, vaginal contractions. They all have to be tightly coordinated because those sorts of reflexes include multiple different organs, secretion of fluids, contractions of muscles, etc. So, and also what's really interesting is that these neurons receive inputs from very important areas in the brain that actually inhibit their output or excite their output. And again, this is a summary of some of the areas. One thing about Barrington's nucleus, which may be important to study in the future, is it is one of those sites that's really important in the afferent information, but it also coordinates and shows up in all the efferent outputs. And maybe, there are very important cells there that help coordinate bladder function on multiple levels. And maybe this is one of the key areas that is involved in crosstalk in the brain. So to conclude, studies in my lab have described the spinal and brain areas that project the different pelvic organs. We've mapped the spinal neurons that are activated with different sexual responses, vaginal distension, erection, ejaculation, etc. So this work provides, be it all small, but it's a valuable information on the central nervous system control of pelvic organ function, which may be useful for understanding and treating dysfunction of not only the pelvic organs, but why impairment of brain sites and neurotransmitters leads to pelvic disorders, as well as how the autonomic and somatic nervous systems interrelate. And we have grand conclusions here that future research in the lab relies on your creative capacity and time and money. And also there are now a lot of new investigative tools that could be useful to help us really further understand the fine-tuning of these systems. So I thank you for your time, and we'll entertain any questions.
Put this all together, what implications do you see for pelvic dissections from surgical technique, common operations, prostate, uterine, abdominal perineum? What practical advice would you give surgeons doing those dissections?
Keep the nerves intact. I'm sure Dr. Carson can probably answer that better. It's a very difficult thing to do because we don't really know where the sub-branches go, even in the human. There's been a lot of recent work done on cadavers. I know especially in the female that really is trying to follow some of these branches over, and they're finding innervation of the pudendal nerve to the vagina, which, be it the distal vagina that wasn't known before. So I think just be cognizant and be careful. And Kelly, if you have anything else to add.
I was just going to ask, that I think this is very elegant, beautiful work, and it really kind of sets the stage for a better understanding of some of the inflammatory disease processes that we deal with and have always sort of treated with surgery, but probably aren't appropriate to treat with surgery. So I was just going to see if you could clarify a little bit more the inflammation in one organ versus the symptoms in the other organ, and how does that work, and what happens if you take the second organ out? Is there still some nerve response?
Yeah. Well, this is sort of a new area of research, at least to me. It's just how does the immune system and the autonomic and the functional system interact? And We don't really know all the answers. We do know that you get excitation of different neurotransmitters and of growth factor. The neuropeptides are released, and they cause sensitization and desensitization of different pathways. If you take out the end organ and leave the nerve, I still think it would be time before you see deactivation. So again, some of these experiments are yet to be done, but I think they're interesting and definitely clinically relevant.
The interesting thing is we've done that in humans with not good results. There was a period of time where people took bladders out for interstitial cystitis and patients still had pain. So I mean, basically this sort of describes the basic science of that, something we do in humans.
Thank you. Any other questions?
This is really great work. It's amazing that in this time and age, we still don't understand basic anatomy and interaction of such an important area. I have a question related to what Dr. Carson had said. That's really great work. And it's clear that you have plasticity and crosstalk between these various neurons. And has anyone looked at an inflammatory condition and how that might, I know that this is just being delineated now, how that might change the interactions and might you have the basic crosstalk, but how does inflammation affect that crosstalk? The second one is a tool question, which is that it's clear that the area is advancing, but is there any way to do any of this in live animals to image the activation of the neurons? I know that's a tremendous challenge.
Yeah, the second one I think is the techniques are not there yet to be able to do that. The first answer to the first question is hopefully if my grant gets funded I'll be able to address that. But there are inflammation has been caused by sort of weird things like acetic acid in the past, just to show the crosstalk and the cross-sensitization or over-distension. We're proposing to use a candida, for example, into the vagina to look at inflammation of the vagina, and then we hope to develop a model that we can look at the bladder function in that same model. But these sorts of experiments are just being done, so I think there's a great opportunity out there to be able to look at developing models of inflammation and how it really affects multiple organs. Because most people work in a sort of sexual function or GI or trauma, and they don't cross the fields. And I think it's really important to bear in mind that you should cross different fields of research.
And that was going to be my last question. So much of the areas of interest that you have are affected by behavioral psychology.
Yeah.
And you show this very elegant connection to the brain. And how does behavior or your interpretation of what these events are, does that go back in crosstalk as well? So I know that would be enormously challenging to investigate, but it's such an important program.
Absolutely. I mean, these common areas keep coming up in our studies, and they come up in other studies. I mean, we're looking at models of depression, and we can see some of these areas are also involved in mediating depression, for example. So again, there's a lot of things that we need to look at, but again, trying to get diverse input. I mean, if you look at Parkinson's patients, one of their early symptoms is bladder dysfunction. So we have to really bear in mind that if we're looking at a neurotransmitter brain effect, we can maybe use some of the pelvic organs as clinical signs, and if we can understand how they're connected, that will help us really sort of help the overall system, which is really important.
In your model, you made a point of saying that you use a sexually active rat. What's the detail there?
So we actually already had these rats left over from a previous experiment, and by coincidence, they had also already been overrectomized. So there was a student in our lab a year or two previous that had done almost an identical experiment, but got extremely little labeling after pseudorabies injection into the vagina. And what we also know is that a lot of these connections are estrogen dependent, and so that all of our animals would they would be on the same cycle. They were all overectomized, actually, by coincidence that we already had them. So.
Yeah, so in that particular study, when we injected pseudorabies virus into the vagina of normal cycling females, we really got very little uptake. When we do it in ovariectomized females, we get uptake. So obviously there's something happening that's causing inhibition of transport under normal hormonal conditions. All right, thank you.